Hello all, welcome to Ranjana's Academy. Today I'm going to teach the topic Sexual Reproduction in Flowering Plants. I'll be discussing the concepts of pollen-pistil interaction and double fertilization. What is pollen-pistil interaction? This is where pollen and pistil are going to interact with each other, or they are going to speak, or they are going to communicate chemically between each other. So this is going to take place in a series of events. We'll discuss one by one each point wise. Okay, so the first point is pollination does not guarantee the transfer of right type of pollen on stigma. You know, stigma is wet and sticky, and pollen is going to come, and any pollen from any family can come and stick onto the stigma, isn't it? So this is called a self-incompatibility. Now, what is the role of pistil? What is something special about it is that it has the ability to identify the right type of pollen on it. So this is called compatible nature. So it will identify, okay, this pollen can be accepted and he can come inside. It will tell the pollen grain. And how does this do this? How does this recognition process take place? By accepting or rejecting? This takes place by the continuous chemical dialogue between the pollen grain and the pistil. Yes, so there takes place the continuous chemical dialogue between the pollen and the pistil, right? Okay, now this is the interaction between them. Now after pollination, what are the post-pollination events? Pollen grain germinates on stigma to produce pollen tube through one germ pore. You know what is a germ pore, right? So through one germ pore, it will extend itself, that is the vegetative part of the pollen will start to develop itself into a pollen tube. This pollen tube grows into stigma, style and reaches ovary. So this is the destination of this pollen tube. So it will come till there. It will provide the nutrients which it already had. Now on the way going, okay, and this growth of pollen tube is chemotrophic. Okay, on the way going, this male gamete, this generative cell, what is generative cell? That elongated piece which is floating on top of the nucleus of your vegetative cell, isn't it? This generative cell will split into two male gametes. This is called as two cell stage. In few of the angiosperms, what happens is there is a different case. What happens is two male gametes will be carried from the beginning. They will split before itself, not at this spot. Okay, that stage is called as three cell stage. Fine, was it clear? Okay, now let us see the entry of pollen tube. So it has come till ovary. Now it has to enter ovule, that is your embryo sac. Only then it can fertilize with your female gamete to form your desired zygote. Okay, now how does it going to enter into the ovule? That is, how is it going to enter into the ovary first? So there are three entries. Just imagine a building which is having three different gates. Okay, the front gate or the main gate. We call this main gate over here as the micropyle. So what is micropyle? This is the micropylar end. That portion is called your micropyle portion, and this is your porogamy. This process is porogamy. When the gametes enter through the micropyle, this process is called as porogamy, and this is where most of the angiosperms follow. They come through the main gate. Okay, now what is chalaza? Chalaza is the exact opposite of the micropylar end, right? So this process of entering into the back gate is called as chalazogamy, and the example for this is Casuarina. Casuarina is the example for chalazogamy. Fine. What is integuments? Integuments are the side sheets, right? They are the thick side sheets. We have two types of integuments, outer and inner integuments. Now this you can compare it to the side gates. Sometimes buildings even have side gates, right? So this is your side gate for your ovule, right? So this process of entry through integuments is called as mesogamy. You can find this in your cucurbita family, entire family, starting from your zucchini, pumpkins, everything will come under this cucurbita family. Okay, now the entry of pollen tube into the embryo sac. So ovary, it has entered. Now we come to the ovule, that is your embryo sac. Always micropylar end will be guided, okay, mostly in angiosperms. Okay, now we know that we have synergids in the micropylar end. What do we have here? We have synergids in the micropylar end, and I can see that these synergids will degenerate at the time when this pollen tube enters. As a result, it will have structures called as Filiform Apparatus, which are finger like. You can see some extensions, some projections here. like this. Now these are the Filiform Apparatus. They are called as the guide. Okay, they are going to guide the entry of pollen tube even further into reach the female gamete, so that they will welcome the male gametes and they will say, okay, your female gamete is here, you're going to go over there. So it's going to show us the direction of the path. Okay, so up to this is called as pollen pistil interaction. And very importantly, you have to know that this is a dynamic process, how the Filiform apparatus guides and how the male gametes and the degenerating vegetative nucleus, that is your vegetative nucleus, after some time it will loose off, it will go off. Okay, that nucleus is about to worn off. Okay, now these two male gametes are entering here, one is going here and the other one is going outer. Okay, why is it so? I'll teach you. Now we are going to see a little bit in extra point. This is one more point, but this is not in included in your pollen pistil interaction. So what is this? In vitro pollen germination. What is this called? First of all, what is your pollen germination? This can be studied by dusting your pollens. See, you're going to take some pollens, you're going to dust them out. Okay, so dusting pollens in a glass side with 10% of sugar solution and boric acid with calcium, magnesium and potassium salts. Any of these salts, you're going to take it in ratios, you're going to put it on a slide and you're going to leave it. So after 15 to 30 minutes, you can see that there's going to be the growth of the pollen tube. You know that pollen tube can be grown only with the chemical language or the chemical dialogues which takes place between the pollen and the pistil. But you can know that even if you provide suitable temperatures in a glass slide, you will be able to germinate a pollen. So you can do this study in your pea, chickpea, crotalidid, balsam and vinca. Okay, so this method is called as hanging drop method. And you know, in vitro fertilization in your human reproduction, yes, test tube babies and all that. So the same process, we are going to put it down into a glass slide and we are going to see the growth of pollen tube. Yes, fine. Now what is artificial hybridization? This is one of the important topic on the basis of NEET as well as in the basis of your board examinations. So what is this? Crop improvement by crosses made between different varieties, species and genera is called as artificial hybridization. Why do we do this? To obtain a single superior quality. See, for example, I want a plant which is disease resistant. It has to live for a longer period of time and it has to yield good amount of fruits or whatever. I have to have a good yield. So if I want all these characteristics in one plant, I have to definitely go for a hybrid, right? So what does this technique involve is, first you have to select your suitable parents. Okay, why? One parent has to be specialized in your disease resistance, another one in your lifespan. Okay, so you have to choose whom do you need as your parents. After that, the bisexual flowers. In bisexual flowers, what are you supposed to do is, you are going to remove the anthers, and this process is called as emasculation. In a female parent flower, you have to remove anthers. Why? Because to prevent self-pollination. What is the reason? To prevent self-pollination. You have to stop this. This should not take place. That is the reason we do this emasculation process. Now, what is bagging process? Covering the emasculated flower by a bag, usually butter paper or some suitable size bag you are using to cover the emasculated flowers. Why? Because to avoid cross pollination. Yes, some other unwanted pollen should not go there and it should not fertilize, right? So you have to bag it and you have to keep it safe. Fine. Now what you are supposed to do, you have to bring your desired pollen grains. Whom do you want as the male gamete? Okay, you are going to bring them. You are going to put it, you're going to dust them on your female part. Again, you are going to rebag them. That is important. If you're not doing so, this before this could achieve, another one pollen will go and fertilize it. So again, you are supposed to rebag until the fruits are allowed to develop. Okay, so you will be giving suitable size. Mentions this, that this fruit should not be affected by the growth of this bag. Okay, this bag has to have the suitable size. If female flowers produces unisexual flowers, there is no need for emasculation. What do you mean by this point? Emasculation is not required. Why? It will already have only one sex. In that case, it is not required. Fine. Now we come to the important second topic of today's session, and this is a simple topic. This one page is the entire content of this double fertilization, students. Two things are involved here. One is the process of syngamy. See, one is the process of syngamy, other one is the process of triple fusion. That is it. That is it. See, what is syngamy? In other words, it is called as fusion. Fine. What happens here? A male gamete and a female gamete is going to fuse, and both are in haploid state. Fine. We are going to get a zygote which is in diploid state. Now, what is triple fusion? One male gamete will fuse with two fused polar nuclei or the zygote to give pen or the primary endosperm nucleus, and this is triploid in nature. Okay, so the syngamy along with triple fusion is said to be your double fertilization. I hope this is very clear for you. This is the entire concept of pollen pistil interaction and double fertilization. Thank you so much for watching.